So what I would like to talk to you today about is a couple of projects that we have been doing recently in our lab and in collaboration with another group on the dynamics of bacterial populations. Let me first begin by thanking the people, because I think it's most important, because they are the ones who did really the work. In the first part of the talk, the experiments were done by Lorena Spina and Marta Diez, who are in my group. In the second part, there is a theoretical aspect, which is in which we work, which was done by Padre, and the experiments were done in the lab of Groswell, who is now in San Diego. Also in collaboration in the mechanical part, the AFM measurements by Conviglu at UT Dallas. Okay, so the main idea of the talk, or the main thing I would like to talk to you about, is self-organization, which in my opinion is one of the most striking properties of biological systems. Of course, people coming from physics, and I see a few of us here, it's not surprising to see self-organizing systems. I mean, non-living matter is also known to self-organize, and in the 1990s there were lots of works in statistical and nonlinear physics showing this type of thing, this type of self-organizing phenomena. But I think we can all agree that the self-organization in biological systems is much more impressive. I mean, the coordination of things happening in a in an organism is really amazing. For instance, one example that some people in the audience have been working for a while is, for instance, developmental biology, and trying to understand how an embryo develops into an organism. And this is, from my point of view, an impressive example of self-organization in space and time, in which you have a collection of cells that, on top of the system growing, you have some mechanisms that provide the system with the capability of growth. And on top of that, you have these cells are growing in number, and then they have to make decisions, and they have to make decisions at the right time and at the right position. And that's, for me, what is most striking about this kind of self-organizing process. How does this work? No? And then a lot of people, some of them in the audience, have really been working hard in trying to elucidate different aspects. And many things are known. I mean, developmental biology is a very well-established field, and there's huge amounts of information. But I think what is still missing is the systems level view, which is, of course, very hard, because I mean the diversity of phenomena happening here, all the different decisions that cells are making, I mean they are happening in different contexts. It's very difficult to comprehend. So then here comes a physicist, and then the idea is, okay, let's try to simplify things a little bit. Maybe the system is a little bit too complicated, so let's go to cells which are simpler, which are also doing decisions, and non-trivial decisions as well. And I like to put this I got from the internet. I didn't prepare these two figures, and it's really interesting that they even use the same background. So it looks very similar. And you see the cycle, the developmental cycle. In this particular case, it's a unicellular population of a slime mold. And then here you have these unicellular organisms that get together and they decide to do different things. They are all identical, but they decide to do different things at different times and at different locations in space. And you have here a very interesting self-organization process too. So the whole message of this second slide is that you can have very simple cells that also have very complex behavior, much more complex than non-living matter. So now we reduce the problem. Of course, it's a big simplification, but I want to understand this type of self-organization. I'm going to do a step further. Maybe slime mold is too complicated, so I'm going to go even further down. I'm going to deal with bacteria. And the particular bacteria we have been working on for a while in the last few years is Bacillus cerealis, which is a soil bacterium, which is very sophisticated in its behavior, because living in the soil, it doesn't have a lot of control over its environment. So this means that a lot of things are going to happen over it, and then the cell has to be ready to make decisions depending on whatever happens. And indeed, that's the case. I mean, for instance, this is just an example of a few different behaviors of this cell. You can have, for instance, situations in which the cells are sessile. They just start growing along long lines, and they don't move much. You have also examples, and these green ones are more motile, and that is the case because they develop flagella. It's the same cell, and it just decides to do different things, and this means that you have to make decisions. So then the question is, how do we decide between being a sessile cell or a motile cell if you have exactly the same genome? Another situation which we have been working also in the past is the case of sporulation. So maybe if conditions in the soil are so bad, the best thing is to hibernate and then you become something completely different. You are quiescent. You don't do anything. You just become a corpse full of DNA. In order to get to this point, you also have to make a complex change of decisions. Again, it's the same type of cell. And there is also another striking phenotype, which is of course very attractive because it's all cannibalism. So this bacteria can cannibalize other bacteria, other cells of the same species, and some killing factors have been established mainly by the group of Richard Losick. And here you can see, I mean, here red cells are dying cells, and if you remove the killing factor, there is no death. In the wild type situation, the green cells are killing the red cells. This is just an example of a very simple organism, and it's relatively easy to work with genetically, so you can do a lot of genetic perturbations. So this means that you can test things, in particular theories, as we will see. It's very simple. You can work with it at many different levels, but yet it has a very complicated behavior. So there are many different decisions that these cells make. Now you could say, oh, but what I can see here is not a very nice multicellular and a very nice population with a very nice spatiotemporal organization. These are just cells probably in liquid or maybe in a solid state, but still this is not. Where is the spatial aspect that, for instance, a developing organism has? Now, the other thing, the nice thing that these bacteria have is that when they get together, they stick to each other, they secrete different types of proteins and molecules that provide an extracellular matrix on which the bacteria can live, and then you can have these type of things. Okay, so this is as close as I can get right now to an organism that has been developed after some stages, and this is a biofilm. Okay, so here you have collections, populations of I don't know, thousands and hundreds of thousands and millions of bacteria working together with a certain very nice, very well established and very reproducible spatial profile, a 3D profile. We will get into this later. So the question is, we can go to the single organism, which is able to do very non-trivial decisions in time, and then we put them together, and then it develops this very nice spatial organization. How does this happen? Let me show you that this is a very, for instance, this paper here was, by the way, this was published in Science. The last author was Steve Shu, who was Nobel laureate in a completely different field, and he was the Secretary of the Energy with Obama the time he published this paper as last author. So this was kind of funny. So the guy, I don't know how he does it. And this is basically a microscopy. I mean, the guy is, he works in optics. So this is a microscopy paper, and what they did is they analyzed a biofilm, not of Bacillus in this case, of Vibrio cholerae, and you can see here in blue you see the cells, and in red and in gray and in green you see all the different components of the extracellular matrix. And what this is telling you is that these little bacteria organize in complicated structures. Okay, that's just they were able to elucidate how is the extracellular matrix organized in some way or another. Okay, so it means that this is a really highly structured in space population of cells. And then the question is, how is this developing? Okay, now, but let me rephrase. So all the questions that I would like to more or less address in different ways today, the first one would be this one. How are the different decisions organized in time? So this means dynamics, okay? And this is something that we, as Xavier was saying, we have been working on dynamics. So the dynamics of cellular decision making is an interesting process in itself, and it's something in which physicists we can say a lot because of our background on dynamical systems and stochastic dynamics and so forth. So that's one question that maybe we can tackle a little bit. How are these decisions organized in time? Another thing that these cells have to do when they are embedded in these populations is you have multiple signals and they have to integrate them. So another question is, how do the different pathways, which are corresponding to different signals that the cells might detect, are integrated and are interacting inside the cell? And I would like to make just address this question in a very specific case. It's going to be very specific. I'm not going to make grand claims because we still don't know, but maybe we do a specific type of a study trying to address this in a very simple situation. Okay, so this is basically what happens in time, and then we go to what happens in space. How is the population level organization determined by this cellular behavior that we were saying earlier? Okay. Now, I probably don't need to sell this to you, and this next 3 slides are I use when the audience is mostly in classical, if you will, biology, in order to motivate why do we need theory? Because after all, I mean, I am a physicist, so we apply our theoretical methods. Why do you need theory in this case? So, and one example that I like to use usually is the following. Imagine this following situation. I mean, all these decisions are going to be caused by genes and proteins that interact in circuits and networks. Okay. So let me put let me set up a situation for you. Imagine these two little circuits. Okay. So we have two genes and their associated proteins X and Y. Okay. And we are going to link them together. We are going to assume that they are linked together in this way. So we have X, which activates itself in a positive feedback loop, and represses another component, which is Y, and Y is activating X. This is a very simple piece, you might think, of a circuit, of one of the circuits that might drive one of these decisions. It happens to be one of the circuits that we will study later, because this is the one in which we have been working on, but let me use it to make a point here. You have this architecture. And let me show you this other one. You have gene X, which again activates itself. You have a second component which now is activated by X, and this second component is representing X. Okay, so these two systems are very similar. This one, X, here you have a positive feedback loop and a negative feedback loop overall, which is mediated by Y. And the same thing happens here. You're just changing the polarity of these two links. Okay. Now my question to you would be, okay, what do you think is going to happen? I'm going to tell you already that, for instance, if you track X in a cell in real time, you're going to see some dynamics. You're going to see some pulses and some things. So, but my question would be, okay, what do you think is going to happen if there is dynamics, or even if there isn't? What do you think is going to happen? What is going to be the correlation between X and Y? I would like to ask you. Let me say again that the two circuits are very similar. What I would like to ask you is, okay, what is the correlation? When X is high, how is Y? Is it going to be high or low? Very simple question. Now you look here, and it's not so clear, because if you look at this arrow, you could say, oh, they have to be anti-correlated, because when one goes up, the other one goes down. But if you look here, you say, oh no, but maybe they are correlated positively, because both, one is activating the other. And then we go in this one, and you have the same argument, but with opposite polarity. Okay? So what this means that biologists usually have these models in their heads about proteins activating or genes and so forth, and they know that in one case you have positive activation, in another case you have negative. What I mean is that when you are embedded in a network, or even a very simple circuit like this one, it's not easy. You cannot say, oh, because I see this positive activation, you are going to see positive correlation. No, now, this means that one way of doing it is you just write down some equations, and this is where people who with a more maybe physics background do it, and down we write some equations. Let me show you first the result. I don't know if you already have in your mind what's going to happen. In general situations, and we have experiments for this. In general, this is not experiments, but we have experiments for this. What happens is, in this particular case, there is anti-correlation, and in this particular case, there is correlation. Of course, you might say this is going to depend on the parameters, and it's true, but this is a very general, it's a very robust kind of behavior. So this circuit implies anti-correlation between these two guys, this circuit implies correlation between these two guys. Okay, and again, I'm telling you, you can check this experimentally, and that's why, even though you make up the parameters in your equations, you more or less can be sure that this is at least existing in nature, because you see it. Okay. So again, we do this because we can write down some equations that describe these circuits. And again, you can try to see whether this happens experimentally. Now, you can do, if you have these equations, you can do lots of things. You can see how things change when parameters change. And this is when then we can play with our model and say, okay, what happens if I increase the expression of this protein over this gene over this other gene? And then you do bifurcation analysis. I'm not going to go into details. This is just a figure that I put here to tell you that we can do a bifurcation analysis and decide and see that in our relatively large region of phase space, the behavior I just showed you is more or less prevalent. Okay? And anyway, these 3 slides were just to convince you that we need this type of dynamical models in order to make some sense of these complicated decisions, because these complicated decisions are, in our cases, going to be caused by all these circuits, in which you have all these different interactions. In some cases, they are contradictory with each other. Okay? So now this means that we have to go to equations. And now the next problem comes, and this is where I would like to come in. When you go and have, okay, I have a cell, I have an input in the form of maybe extracellular signals, and then I have an output in the form of a phenotype. And then what I do is I write down some very simple dynamical model, a collection of ordinary differential equations. For instance, to begin with, we're looking at what happens in space, that tell you how the state of the system defined by this vector changes in time as a function of the same state of the system, and some external parameters, which are the inputs. Very simple. Okay, so you have here a model, and then we have inputs, outputs, and interactions. That's it. Now, the problem is that in many cases we don't know what the parameters are. It's a very complicated system. I mean, it's not only two genes. So, and we need, in order to make a good comparison, we need to quantify. Our problem, we have a need of quantification. We need to be able to compare the model with the experiment and not leave the comparison to arbitrary things. I mean, I want to make an absolute comparison. That's one of my goals. Okay? So we need to quantify and compare absolutely inputs, interaction, and outputs. Okay, what I'm going to do today is I'm going to concentrate on, I'm going to show you, I mean, answering the questions I was telling you earlier. I'm going to use them as an excuse to show you what we have done, trying to be a little bit more quantitatively in comparing the inputs and the outputs of these circuits between model and experiments. Okay, that's the whole thing. I'm going to leave the interaction aspect to another, I mean, I have other examples, but I didn't have time, so I'm going to do these two things, okay? And then I'm going to use model behavior in Bacillus, and this is a behavior in which we have been working on already for years, but we know it a little bit, which is competence, okay? Genetic competence. Genetic competence is a phenotype in bacteria that allows the cells to uptake DNA, and this gives them hopefully some chance of survival in a hard environment. Okay, so of course genetic competence is used a lot in genetic engineering because that's a way of getting DNA, external DNA, to the cells. So this means that people have studied for a long time, and what we have been seeing in the last years is that this is a dynamic type of phenotype. What you see in red is the cell, the competent cell, and green are just stressed cells. Okay, so you can see here that the phenotype is strongly dynamic. I mean, there is a decision. This cell here decides to become competent, irrespective of what the others do, if you will. But this decision is transient. I mean, it stays there for a while. We have reasons for that. I'm not going to enter into this. I just want to emphasize this behavior because it's nice. It's a nice post-like behavior that we can analyze. And we have done this. And now I want to use this as an excuse in order to, I mean, we know a little bit how it works. And the main circuit behind it is one of the two I was telling you earlier. We have one factor, which is the master regulator of this decision. This is COMK. This is the master regulator of competence, which activates itself and inhibits another gene, which is basically the stress detector. And this stress detector activates COMK. So it is the circuit that I gave you on the left, in which I told you that you have anti-correlation. In fact, you can see that there is anti-correlation because cells are either green or red. There are no yellow. I mean, the cell is not yellow. So this means that when green is going down, red is going up. So you do have anti-correlation in this circuit. Okay? Now, we have been working on this for a while. We have been validating. I mean, we have a mathematical model for this, and we have been validating it. But there's always the problem of what happens with the parameters. The comparison, I mean, we have never been able so far to do a quantitative comparison in absolute terms between the model and the experiments. And what I would like to try to do is a first step in the direction. Now, how do we do it? It's relatively simple also now, but the question is, okay, we have to do it in a careful manner, and this is, I'm going to show you. First, the inputs. I'm going to apply inputs to this system. And I'm going to try to address the question of integration. So this means that I'm going to consider two inputs. And I want to see how do they integrate. So my case example is I want to understand how this circuit integrates two different types of inputs that are applied to it at the same time. And the inputs are going to be, the first one is nutritional stress, let's say, which is the natural input for this circuit. This circuit activates when the cells are stressed because there's no food. And the stress is known to enter the circuit through this, because I was telling you earlier this is the stress detector. So this is our first input. Hopefully, now I'll talk to you later about it. Hopefully, they'll try to control it because we want to make a quantification. And then another inputs, and this is a little bit more, if you will, artificial, but it could be important. I think it is important, for instance, in developing organism, which is I could have a constitutive expression signal. I could force into the system a constant expression of either this gene or this gene, and this is going to be an input in my circuit. Now, this could be interpreted as a context-dependent signaling in development. Okay, so you have your system, and then depending on what this input is going to be, the circuit is maybe going to respond in one way or another. So that somehow these inputs, which I'm going to consider separately, modulate the response to the stress. But then I want to play with them. Okay, how is this little circuit integrating these two inputs? And now I'm going to first, now let me remind you, we will have a model for this that I'm going to briefly show you. And the goal is, that's very nice, but we need to be able to quantitatively, in an absolute way, quantitatively compare inputs and outputs between theory and experiments. Let me begin by the output, okay? Now this circuit is good for this type of thing because it's dynamic, okay? It is a dynamic behavior. I mean, cells go in and out of competence, okay? You have a cell here and then eventually it turns on competence. There, I was telling you there is anti-correlation because the comments gene goes down. And then you have here a clear anti-correlation. You can see it very clearly. But the important thing for me is that you have, this is a dynamical event, and I can characterize it in a way that it's independent of, for instance, what is the efficiency of the fluorescent protein. One problem that we have when trying to make comparisons is that this protein, I mean, it has really not so much to do with the original protein, and the model is telling me I want to know what is the state, or what is the level, if you want, of expression of the protein, but this is, I'm not measuring this directly. So one way of getting rid of arbitrary factors in this comparison is by making use of the fact that this is a pulsed dynamical behavior that I can characterize statistically. And for instance, what I can do is, okay, I see an initiation event, I see an exit event, I see maybe a reinitiation. If this guy is oscillating, I can do experiments and I can compute the statistics. And these statistics are going to characterize the output of the behavior in a way which is independent of arbitrary factors related to the fact that maybe the different colors, YFP and CFP, how do you call it? Maturate at different rates. And then this means that I cannot compare the two. So I really don't know if really Y is being expressed more strongly than C. So this means that I cannot compare this with my model. Let's forget about this. I'm just going to do a statistical comparison. I just want to see, okay, I measure the probability of initiation, the probability of exit, and the probability of initiation. And then since it is dynamic, then I can associate, using these probabilities, I can associate this with dynamical regimes. For instance, if probability of initiation is not too high and exit probability is high, and the initiation probability is high, this means that I have oscillations because I enter relatively quickly, I exit very quickly, and I enter again very quickly. So this means I have oscillatory regime. I have other situations in which I could have bistability, for instance, in which I enter and then I exit. I don't exit so often. So this means that I get stuck in the other state. So this means that I have two stable states, for instance. Anyway, we can go through all of this. The important thing is that you can really make a table and you can say, okay, for this parameter, I have this type of dynamical behavior. And in that sense, since I am using probabilities, and not using absolute levels of expression of a given protein, I consider this to be absolute. And if my model tells me that for a given input signal, I have to be in oscillations, there is no arbitrariness. I mean, the experiments have to, I mean, if the experiment doesn't show oscillations, this means that my model is wrong, okay? Now, you could say, oh, but this is not really, I mean, this is just too abstract or too qualitative again. Now, the thing is that you can do bifurcation analysis and decide when you're going to have changes between the different regimes, and this is going to be quantitative, okay? So that's the way we quantify the output. Let me go to the inputs. How do we quantify the inputs? So let me remind you, these are the two inputs that I'm going to be considering. Let me begin by this one, okay? Okay. So what I'm going to do in order to include some expression signal, some basal level, some constant level of expression, additionally on top of the natural regulation of the circuit, is I'm going to use an inducible promoter, which is this one is called hyperspank promoter, which is a promoter that you activate externally by using IPTG, for instance, some chemical that you add on your medium. And this promoter is going to express, for instance, CAM-K. So this means that I can really control. I use my chemicals, and then I can control how much CAM-K I'm producing, and this production is constant. So this is going to be a term in my equation that corresponds to a constant production of CAM-K. And we can do this because it's relatively easy to work with bacillus and introduce this construct into a cell, and then in that way you are perturbing the circuit. Another input that we can do separate is the same thing, but acting upon this. Okay, so we can do the same thing. So this means that we have two knobs. We cannot turn the knobs, these ones cannot be turned simultaneously because in particular we have the same promoter here. This doesn't mean that we can even include a second inducible promoter different, which is not There is no crosstalk with the first, and then we could introduce these two things simultaneously, but we haven't done it. But it would be possible by just using a different inducible promoter. Okay, that's one thing. But then we have the problem of quantification, because my control parameter is the amount of IPTG I put in the medium. Okay, and of course, how do I relate this with my model? In my model, what I will have is just some rate of production of CAMK. How do I relate the rate of production of CAMK with the amount of IPTG, I don't know, 1 micromolar IPTG I put in my medium? It's very complicated, and we cannot do it in this because there are many unknowns. So how do we do it? Well, what we can do is normalize. I mean, what we do, for instance, is we measure, for instance, what is the effect of this input with respect to the natural expression of the proteins. For instance, if I'm including here IPTG, which is going to generate a constant amount of CAMK, what I do is I monitor also CAMK in natural conditions in the cell. And I do this by using the same color. Okay, now I'm using the same fluorescent protein, which is driven by a promoter, which is a reporter for CAMK. And I use this to normalize. So this means by comparing, by doing some, I just do, I have two strains, one containing this, one containing this. I start adding IPTG, and then I compare the signal I get from the IPTG promoter with respect to the natural signal, the fluorescent signal I get from CAMK. And then this means that I'm going to measure the result of this input in units of the natural basal level of production of CAMK. So in that way, and you will see that in a minute, I will show you I should have maybe shown you the equations already, but maybe you already have it in mind. So the idea is, by doing this, I just normalize. I just say, okay, when I have 1 micromolar IPTG, what I'm doing is I'm introducing into the cell an amount of CAMK which is 2 times what the cell is generating naturally when it's not competent, when it's not in the peak, okay? When it's in the basal condition. So I can say 2 times or 3 times, and I have it here, 10 times, 20 times, and this number, I can compare between experiment and model quantitatively in an absolute way, okay? So, and this is what we are going to do. Now, there's no arbitrariness, because I'm just doing this. So, and the same thing here. You can do the same thing for CAMS, okay, for this one. So what you do is now, you do the same thing. You are able to tell, okay, when I'm adding here how much IPTG, I'm going to tell you how much is it in units of the natural production of CAMS in the absence of competence. And these are parameters that I have in my model, and I can really introduce and compare. Okay, so that's my way. This thing here is already allowing me. These are going to be the. I mean, it's only 4 data points. Poor Lorena here had a terrible time because it's a lot of data that she had to process. So we didn't do anything more than 4 data points here, or 5. Well, besides the wild type. Okay. So anyway, let's go to the second one. This one is more complicated. I'm a little bit slow, but okay. So this one is more complicated. Why? Because stress. It's complicated in a situation in agar conditions, the ones that you use in your microscope in the movie that I was telling you earlier. The stress, you cannot control it. The stress is growing because there are more and more cells. Cells are growing up. They are emitting signals. So in the medium, then the amount of stress signals that the cells themselves emit is growing. So it's very difficult to control. Okay. So this means that if I want to quantify this, it's complicated. So let's do a trick. Okay, since this is a stress detector, I'm going to mimic a stress by having multiple copies of this gene. Okay, so now what I do instead of the stress, I'm going to do a copy number perturbation. What I do is I add multiple copies of this gene in such a way that if I have 2 copies, I'm going to say that this is maybe twice the stress at any given time. So, okay, or I can have more. And I can do this very easily because I can use plasmids, which are available, and more or less it's known what is the copy number, more or less. Okay. So what I'm going to do is I'm going to introduce extra copies of the natural gene, for instance, in plasmids. And there are plasmids, which are little chromosome DNA rings that behave like a chromosome. And in some, so usually these bacteria have 1 copy of the chromosome. And if you have a certain plasmid over there with another origin of replication, it's going to be replicated as the cells grow and divide. And maybe you're going to have plasmids on copy numbers of the order of 10 or 20, 100, and this is more or less characterized. This means that you know these numbers more or less. Of course, more or less, we have to quantify it. So again, we use the same type of very simple technique in order to quantify this. Okay, so what we do now is we introduce, again, I'm telling you, this gene in the plasmid, And we use low copy number plasmids and high copy number plasmids, for instance. And then what we do is we measure the signal, fluorescent signal, which is produced by this promoter once it's in the plasmid, and we compare it again with the signal that is produced when the plasmid is not there and the signal is only coming from the single copy that you have in the chromosome. In that way, now you divide, and again you have how many times you have the natural. I mean, when you have, I don't know, this gene in a given plasmid, this will correspond to having the parameter that corresponds to this regulated expression of this. It's going to be multiplied by n times the one in natural conditions, where n is the number of copies that you have. Now, how do you characterize the number of copies? If you don't want to believe what comes from the characteristic sheets of the plasmid when you buy them, or when another researcher gives it to you. For instance, there is one plasmid that we got, which is a 6-copy number plasmid. So it is more or less known that this plasmid, when it's inside the cell, there are going to be 6 copies of it. Okay. So what we did is we did this thing in order to see whether this was true. Okay. Do we get 6 times the signal? And what we did after, what we got after some, and this is the fluorescence in time. You track the cells and blah blah blah. This is one specific example. We did, of course, the statistics. And after some transient, what's happening at the beginning, which cells are, I mean, at the beginning, cells were in liquid, and then you put them in the microscope. They don't know where they are, so and they have to settle. So I'm going to ignore what happens in the first 1,000 minutes. What is interesting is that we saw, well, in this particular case, is that after 1,000 minutes, the level of expression more or less stabilizes, and we saw that it stabilized around 6.5. So this means, oh, good. And we did the statistics. So we said, okay, well, copy number of 6 that we got from whoever gave us a plasmid is more or less reasonable. So this is one copy. This is another input that we are going to use. We will have the wild type, and we will have a situation in which we have one parameter that is 6.5 times the wild type. Then we get another one which was a 20 copy number. So the researcher that gave it to us estimated, and this is going to be in 20 copies. Now we did the same thing and we got 75. So that's the number that we got. That's all we can say. So we're going to stick with 75. Okay, we measure the number. This is one way of measuring the copy number, if you will. So anyway, these are the 2 parameters. So we're going to have 3 values of this input. 1, which is the wild type, 6.5, and 75, which are the 2 perturbations. So this means that we are going to be able, now I'm going to go back, we're going to be able to have these 2 perturbations together, this and this, and this and this on the other side. So we have 2 pairs of perturbations.
And you are always using fast degradation tags.
Yes. For all our fluorescent proteins have the fast degradation because we're looking at the dynamics. Yes. So the fluorescent protein is not being accumulated. I mean, as soon as it's produced, after a relatively short time, it's going to be degraded. So this means that when you see a signal, this means that it has been produced relatively recently, and you're not filtering out. Yeah. Okay. So then the thing is, now we're going, for instance, let's going to take this pair, this and this. Now we're going to have 4 or 5 data points of this and 3 data points of that. So we are going to be able to map the behavior of the circuit in a 2-dimensional parameter space. That's the thing. And we are going to map it from the model and from the experiments. Let me tell you let me show you the results of the experiments first. And this is a lot of work, this figure. I mean, for Lorena, it was really terrible because she had to analyze hundreds and hundreds and hundreds of movies. I hope she doesn't hate me for that after that. I'm not sure. So anyway. So, for instance, the initiation probability. What we see here, you see 3 colors corresponding to the 3 different copy numbers, wild type, 6.5, and 75. And then you see here the 5 data points corresponding to the 4 levels of IPTG and the wild type. Okay? Well, in this case, I think it's 5 levels of IPTG, and here you have 5 levels also on the wild type. Okay? So anyway, you see here that we are varying the 2 inputs simultaneously. And what you see, for instance, is that when you vary alpha S, I'm sorry I'm not introducing this, I should have introduced this now. So this thing here is going to be the IPTG level, the constitutive expression of COMES. Okay, so I vary it. And this thing here is a 3 plasmids. So you can see that when you vary the constitutive expression of COMES, the initiation probability doesn't change much. When you increase the copy number, it changes a little bit, but it stays flat more or less. Interestingly, for instance, the exit probability at the beginning, it's high. This corresponds to the excitable regime, but after a while, cells get stuck. Okay, and this corresponds to a bistable regime, because now if you do your table and thing and you divide, okay, depending on what are the values of the probabilities, I'm going to decide which dynamical regime. And we can do this for all this. Okay. again, this is what we apply. Okay, we apply this table, we filter, and we say, okay, now I can have my two-dimensional parameter space for the two pairs of perturbations, and this is the results. These are the results of the experiments. Okay. Now, what I want to emphasize is that the axes in this diagram and the colors are absolute. I mean, there is no arbitrary factor there that you include. I mean, these are absolute. I mean, the model, and this is a strong constraint for the model. If we wanted to code, I mean, this is strong. I mean, because we are saying, okay, you just multiply this factor by this fraction, normalize. So this is something that you do, and there is no way out. You cannot invoke some parameter you don't know. That's what it is. Okay, and this is what we get in this case, which is Comes copy number, Comes expression. You have a situation in which there are a little bit of excitability, and the rest is bistable. In this case here, you have K expression and Comes. Here you have a little bit. It's more fun because you have all 4 different dynamical regimes. Okay, that's what we see. Now I'm not showing you movies. Yes.
Just a question, but it's difficult to compare your results that you get from the COM-K and COM-S because one of them contains a plasmid, and the plasmid by itself, they change completely the metabolic state of the cell.
Well, we are ignoring, yeah, that's a
lot. Also, if we compare 6 copy compared to 75
to
copy,
75.
this completely
well,
makes
this is a possibility, no? We have to.
It's well known that one.
Yeah, but the question is whether this change in the metabolic state is going to impact the specific response that we are.
Incompetence for sure.
Well, no, but the impact that we are, at least the impact that we are seeing, we do see an impact. Okay, this is what you see when you increase the copy number. These are the 3 situations. You do see an impact. That's the whole point. I mean, these are experiments, I'm telling you. And the impact that we see is that what was excitable here becomes stable. What was oscillatory here becomes unstable. So this is the impact that we measure. I agree with you, then this has to be, I mean, the question is the circuit that we have, very simple, terrible simple circuit, is enough to explain the same, is it going to, I mean, analysis of the circuit, mathematical circuit.
But you're introducing a really perturbation in the COM-K.
Yeah. But with
just
this,
only for the plasma.
Yeah. But this perturbation we include in our mathematical model. Again, all models, as my experimental colleague told me, he always tells, and I agree with him. All models are wrong. No, so the model is going to be wrong.
You
The
can
question
correct just only cloning the plasmid without anything in your.
Oh yeah. And this is I agree with you in order to check. But this is something that we haven't done, and this is already being planned. Okay, but what I'm just telling you is that we are measuring the effect. Okay, and now we are taking a very simplistic model and we introduce the extra copy numbers in our simple circuit. And this means that we are ignoring all other things that may be important. Okay, that's true. But now the question is, well, let's see what the model says, given the approximations. And again, the model is going to be wrong in all cases, but let's see how far it goes. That's the point. That's what I want to say. I mean, I'm sure I'm not going to say that this is the mechanism and that's all, because otherwise we only need maybe 20 genes in a cell or so. So, and I admit that it's going to be wrong, but let's see how far it goes. That was of course, I'm explaining this to you because it's going to work well. I mean, the simple model is going to agree with the experiments, which means that maybe all the effect of having extra copy numbers are already embedded in the model, and really all the extra metabolic load is really not impacting so much at the level of the model that we're considering. Maybe the main impact of the extra metabolic load is on the model that I have, which involves only 3 elements.
You will see the frequency of competence.
Yeah. You know that, and we compare. And we use it, and we really compare this between model and experiments. We did already some time ago, and this already more or less. And now this comparison is qualitative, but the qualitative comparison also holds more or less. Now I'm telling you, this one is more quantitative, because you are saying, okay, hey, the change is going to happen between 1 and 10. Of course, I'm not doing. Probably, if I did a more careful analysis and used many more plasmids and had it. Maybe we would see differences between the model and the experiment, because the model is not complete. But at the level that we are doing, I'm going to show you now that it works more or less. Okay. Now let me show you the results, because so this is the model, and these are the parameters. So we are changing these two parameters separately, and we are changing this parameter. And this is where the copy number enters. So I'm introducing the effect in the copy number by playing with this parameter here in a very, very simplistic way. Okay, I admit it. But that's it. So now what we do is we do bifurcation analysis and all these type of things, trying to see what are the different dynamical regimes that we see. And that's what we see here. Okay, now what I'm showing you in this plot is the following. These three graphs are exactly the same in terms of what we are plotting in the axis. This thing corresponds to COMES copy number, COMES expression, and this corresponds to COMES copy number, COMK expression. These are the two experiments. And here, what the difference is between, I'm plotting in grayscale the initiation probability, the exit probability. So in that case, we are measuring the frequencies. I'm not comparing quantitatively the frequencies. I'm just comparing the outcome. What does it mean having this frequency together with this a certain exit probability and a certain initiation probability? Okay, so we do this with stochastic simulations, and we can also analyze, do a bifurcation analysis of the deterministic model, and we find different kinds of boundaries that separate the different regimes. We see saddle node bifurcations, we see hop bifurcations that separate the different regimes eventually. So that's what we have. We have excitability by stability, and you can already see that the system behaves. I mean, this is the prediction. This is what comes out of the model. And if you remember the results, the experimental results, it seems to work. And if I put them together, indeed it works more or less. Of course, again, we have to define the different dynamical regimes in a certain way. There is a certain degree of arbitrariness, if you will, in the thresholds that you use to define your dynamical regimes. But on the other hand, analyzing the stochastic simulations tells you that really the behavior is kind of robust. So you can change the threshold quite a bit, and then this dot would become green if you use a little bit different criterion. But when you are here, it's very difficult to decide. The important thing is that on the whole, the system obeys more or less what the simplistic, terrible simple model tells you. Okay, and it is quantitative because you are varying the parameters, and you are doing it in an integrated way. You are varying the two of them at the same time. Now, what do you see? For instance, okay, what we see here is that well, let me show you time traces, so that at least I'm not going to show you movies because the movies are quite noisy, but I'm going to show you time traces. Okay, sorry, let me go back. I'm going to show you one example here and three examples here. Okay, 1, 2, 3, 4, and the same thing here, 1, 2, 3, 4, so that you can see all the different dynamical regimes. And this is what we get. So that's to have an idea of all the effort, because we have to click on these cells, and we are using custom-made software. We are not using, that's another issue. So Lorena was tracking all these cells, computed the histograms, and you can really see all the different types of regimes. When it's bistable, you do see a separation. You do see cells that are never becoming competent, cells that are becoming competent and state competent. And these three cases, the same. This happens in the three cases. When it's excitable, the cells, when they enter into competence, they come back. So it's excitability. In this particular case, the same thing. You have bistability. In this case, you do have monostability. All cells enter competence and become red and stay there. This is excitable again, and this is oscillatory. This is just, sorry, the dark lines are just single time traces. These are not averages at all. This is just to see that in one case, this is one oscillation. You have many oscillations with different phases and so forth, but you do see oscillations, okay? So this means that, well, there is a relatively good comparison between modern experiments, and we are analyzing the integration of signals, because now what I'm doing is, okay, what happens when I try to integrate two signals? And I'm going to go back to this in a minute. Just one more thing. You could say, and it's true, this input, this COMES copy number, is not real. I mean, this one is definitely not real. It's artificial. I'm mimicking some kind of context-dependent signaling, the IPTG input. This one, it's also artificial because, I mean, I'm just introducing extra copies of the gene inside the cell. And I wanted to mimic stress. What happens if I put real stress? Now, if I put real stress, I cannot control it very well. I was telling you earlier, because I cannot say, oh, I'm going to do it. It's not so easy. So I'm just going to do it qualitatively here. I'm going to say, I'm going to use another medium with high stress. Okay? I'm going to sit here. I'm going to sit here. And I'm going to, instead of increasing the copy number of COMES, I'm going to use another medium, condition medium, which is more stress. It corresponds to a higher level of stress. Okay? And I'm going to see if this goes into bistability. And this, I'm going to go to this one, which is, okay? I'm going to take the cells that are operating in an oscillatory regime, and instead of increasing the copy number of COMES, I'm going to put them in a more in stronger stress conditions and see whether I get monostability. trying to mimic the real signal that we cannot control so well. And indeed, so that's what I'm going to do. I'm going, well, in fact, sorry, I was not doing this one, but this one, okay? So I'm just going here and increasing the stress, here and increasing the stress. And that's what we get, okay? I'm just going to, I'm just showing you these two, where I have not introduced, now I don't have extra copy numbers and extra copies of the gene. I'm just having a larger stress. And that's what we get. We do get the bistability in one case and the monostability in the other case. Okay? So this means, now go back, this is something also relevant for biology because there is some debate in the literature whether competence is excitable or bistable. Some groups see bistability or say they see a bistability, and some groups like us say that we see excitability. Maybe it's just a matter of the amount of stress the cells see. What we are seeing is that, okay, if you have a little bit more stress in your medium, you're not going to see excitability, you're going to see bistability. So this is also explaining this apparent discrepancy that we see in different studies. Okay. So I'm going to finish with this. I'm sorry about the biofilm but anyway, I'm going to. So I just wanted to tell you, okay, what are the physiological consequences of all these things, besides all the idea of having this quantitative comparison? The consequence is that, okay, now I want to see how the system integrates signals. And for instance, in this particular case, integration is quite trivial. If I apply a signal, this is a wild type, I apply a signal here, I get bistability. I apply a signal here, I get bistability. I apply the two signals together, I get bistability. So it's kind of trivial integration. This one is more interesting. I have this signal, I apply stress, and I get the bistability. Here I apply oscillations. I apply the two now, and then the sum, if you will, bistability plus oscillations means monostability. Now, physiologically, it's important because for me this is a lethal phenotype. I mean, because both excitability and bistability allow you to have spores, which is at least a way of for the cells to survive. Okay, if the stress is very high, at least they will always be hibernating. Okay, so this is already something, and you can still have vegetative cells. Oscillations still is also a way of keeping. You still have physiological competence because cells go in, absorb DNA, go out and do their stuff. Go in again, absorb DNA, go out, do their stuff. So here you still have the physiological state of competence. Here you have nothing because since the cells are stuck in competence. These cells that are in competence, they cannot do anything. The poor little guys are going to die because they get stuck there. They cannot divide. They just wait for their death if they are stuck in competence. So this means that really the phenotype is, I mean, after a while, there's going to be no cells. So that's so this means that in this particular case, this integration is lethal. That's the most biology I can tell you because it's very. But that's the biology that the physicist does. Okay, that's not. I'm getting, and that's all. Well, a physicist like me, I think so. Anyway. Okay, let me go in the last 10 minutes. This is, now I know I was late, but
you can use them.
So, or 5. Well, let me go very quickly because I mean this is. So now the question. Let's go to the population level organization. Let's go to the biofilms now. Okay. Now let's put together all these cells. I haven't told you about the sporulation, but in this particular case, for instance, sporulation plays a big role. Because this is one of the decisions that cells make. Of course, on their way to biofilm, cells are going to become competent, blah, blah, blah. So the question is, how do cells form this structure? This is very interesting, again, for a physicist even, because, okay, this is very nice. You have these three-dimensional structures. These are very little nice wrinkles. Cells are moving up. You have valleys, and there are different physiological advantages. I mean, these wrinkles in 3D make the biofilm highly impermeable, for instance, to antibiotics. Okay, that's one thing. A group in Harvard says, David Baitz's group in Harvard says that now these little wrinkles provide channels under the biofilm and through which nutrients could get into the center of the biofilm. This will be like angiogenesis type of thing that you have in tumors. In fact, the group of Herbert Levine says that these bacteria have this type of biofilms. Somehow they are linking it with the behavior of tumors, which is a bit crazy, but the idea is that the cells have to divide, the population has to devise a way of getting nutrients inside it. And the group in Harvard said that this might be through these little channels. Okay, so this type of 3D structures have physiological relevance. Now, how do they form? Now, here I'm going to show you the beautiful experiments of Munehiro Asali from Grosswell's lab, in which we show here the biofilm, and then he does experiments. Okay, he does experiments in time at 0 hours, 12, 22, and 46, and you can see here how the biofilm develops. I'm going to show you a movie in a few seconds. But now, what is the interest? The interesting thing is that he can monitor cell death. The question is, he happened to have a dye for cell death. cytox, which is a dye which, when the cell is dead and then the membrane becomes porous, the dye enters the cell and then marks the DNA, and then you see it fluorescing, or if you we use fluorescent cytox in green. So when we see green, this means that cells are dead. Okay, so then what he saw, he did amazing experiments in which he did cross sections of the wrinkles. Okay, and then this is a wrinkle, and then on the bottom he sees cell death, a pack of cells that are dead. If you look at the electron microscope picture, you see here cell deaths, and these are alive. So really, there are packs of cell death on the bottom of the biofilm. Okay. Now the question, of course, is what is the cause and what is the consequence? That's basically the question that we would like to address. Is a cell that causes a wrinkle or vice versa? Okay. Now let me show you the dynamics, because he can measure this, but he can also measure the dynamics of death. And this is the dynamics of death. Okay. Here there is no more death signal anymore, but there is here a lot of death. You can see here you have in the ring, due to the coffee ring effect, cells have been moving to the outside. I mean, this is a drop. You put the drop and cells move to the outside, and then you have a lot of death here because you have a lot of cells. But in the middle, you have little patches. Okay. And then again, a physicist sees this and becomes excited because there's a lot of dynamics. In fact, let me show you these two things side by side in a movie. Okay. Now, this is what happens. Here you have the wrinkles in right field, and this is the cell death. And you can see here the pattern of cell death. And you can see that the pattern of cell death is highly dynamical. And you can see here the wrinkles. But if you look at, you see little blinks, pop, 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 pop. If you track them, there are little areas in which, so cells, there are some cells, some local areas which decide to die later. That's the whole point. There are areas in which cells die soon, and then there are areas which decide to die later. In fact, you see here a lot of death, and then there are some holes, and then these areas die later. Okay? There's a lot of dynamics here. And this is correlated with the wrinkle formation. Why? How? So that's the question. Now, let's look a little bit more closely to the dynamics of cell death. This red here means there are cells, and again, green is the cytox signal. So you can see this is a beautiful example of a spatiotemporal organization. Okay, this is a pattern in time. This is a temporal pattern, it's not stationary. How does this pattern emerge? So our first goal, before trying to answer the question, who comes first, death or wrinkle? Okay? The first question was, okay, how does this pattern arise? Maybe if we see that the cell death pattern arises well before wrinkles happen, this means that maybe death is the one that's causing the wrinkles. So let's write a little simple physicist type of model of this system, a spatiotemporal model, trying to see whether we can account for this type of a spatiotemporal pattern. And the model is relatively simple. We knew, well, this is after our nth trial of different types of models, so this is okay. So we decided to focus on the density of cells and the fact that we have an extracellular matrix I told you earlier. And we draw this little diagram in which local density growth, there's a growth that increases local density, and then there's production of matrix. The matrix is limiting the expansion because when there is a lot of matrix, cells cannot move, okay? And the matrix is also, so this means that you are increasing local density because cells cannot spread, okay? And then of course you have local stresses that kill the cells. And you draw a very simplistic model in which, for instance, the local dynamics is more or less a logistic equation, and then you have diffusion of the density that corresponds to growth of the colony, in which the diffusion coefficient depends on a quantity which is the matrix concentration. So we have another variable for the extracellular matrix concentration. So the question is, okay, what is the matrix? So let me tell you first about the logistic equation. We have a carrying capacity here, and in this carrying capacity, I'm not going to go into details, especially because I'm already late, but just very quickly. So we have a carrying capacity, and this carrying capacity decreases. There is a term that decreases due to nutrient depletion, because of course nutrients are going to be depleted, and nutrients diffuse very quickly, okay, inside the biofilm. So we just assume that there is nutrients going down, down, down. Eventually, since there are a lot of there are not so much nutrients, you're going to have a lot of death. So that's what's killing all the remaining active cells. And then we also have waste. We assume that cells produce waste. We produce signals that are eventually also going to produce delay growth. And finally, we have the matrix here. So we have a diffusion coefficient that depends on the matrix like this, and the matrix, we assume it grows with the density of cells in this manner. Okay, we have lots of parameters. So again, we come back to the problem of how do we compare? But okay, we use more or less reasonable values for the parameters. That's all we can do, and then we look at the sensitivity of this for the specific parameters that we chose, and this is not very sensitive. Let me tell you what happens. So, for instance, the model already gives you, this is a representation that separates early death events and late death events. Okay, so these ones are early death and these ones are late death. And you can see here that you see the same type of pattern in which there are holes in which cells are not died and then they die later. Okay, and so there are regions in which you have death is localized. Okay, so the question is that we see, and now you will see later how this impacts wrinkles. Okay, before that, let me show you. We tested the model in one way. We tested the model and say, okay, if the matrix is so important, let's assume there is no matrix. Let's assume there's a strain of Bacillus which is not able to produce a critical element in the extracellular matrix, which is EPSH. So we have an EPSH mutant, which means that we're not going to have this. Of course, what happens is there's no pattern at all. So this means that the matrix, the term due to matrix production is really critical in order to get the pattern. So what we did is we tested this experimentally in a large way. I mean, Munehiro did the mutant, an EPS mutant. He not only did one, he made 30 mutants. So again, amazing amount of work. I'm just showing you one situation, which is the one for EPSH. I mean, if you look here, this bar here is just a measure of how complex the pattern is in space and time. Okay, and wild type is here. So you see there are many mutants that are not affecting the complexity. All these mutants are not affecting the complexity. The main mutants that affect the complexity are down here, are the matrix production especially. And down here you have EPSH. EPSH, and these are experiments, give you no pattern at all. Okay. Of course, that as far as a validation of a prediction we go. Okay. I'm not going to claim that this is the proof that the model is correct. In fact, we are now trying to disprove the model, okay? Because we see other things that are not. But for the moment, at least this aspect is important. Matrix is crucial, okay? Now let's go to the physiological importance, and I'm done, okay? The thing is, why is this cell death pattern important? So the first thing is, I'm going to show you again. This is a picture in which you can see the three-dimensional structure of the biofilm, and you do see the valleys and the wrinkles and so forth, okay? And let me go back to the assumption. Our assumption is, since we see the cell death pattern appearing first, our assumption is that, okay, cell death pattern appears, and what the cell death pattern is doing is it's changing the mechanical properties of the biofilm in such a way that there is stress, there is compressive stress in this direction, and this is going to lead to the wrinkle. That is our assumption, okay? And so you can see here the cross sections. You can see how the cell death is down here, and then you see the wrinkle appearing. Okay, so indeed, cell death pattern, which we are showing with the model that it only requires the dynamics of the matrix and the density. Then you have the cell death pattern, and then on top of that, this alters the mechanical properties of the medium, and this produces the wrinkles. Okay. Now, is it true that you have this type of movement? So what we can do is we can do an experiment with fluorescent beads of the whole biofilm, and then we can track how the beads move, and this is tracking the movement of the cells, if you will. And so we can track here all the different motions, and we can plot a convergence plot. Okay? And this is, again, so this would be a pattern of convergence, and you compare this with the death, and you can see a pretty good correlation of the areas where there is death with the areas where you have high convergence. So really, the population is moving towards the areas where cells are dead. Okay? Okay? So in fact here, if you can superimpose the pattern of convergence and the wrinkles, and you see a good correlation. Okay? Now this means that this matrix is changing the mechanical properties of the tissue. So we tested also this. I mean, this requires that the matrix component change the mechanical properties of the tissue. And we tested this by comparing, for instance, the wild type cells with cells in which you remove a critical component of the matrix, a different one. We couldn't use EPSH because EPSH we don't see anything. So it was too large perturbation. So we use SFR8, which is making the matrix a little bit softer. I mean, the tissue is a little bit softer. And you can see it here. You can look at the stiffness, and here stiffness is smaller. Wild type is here, and this one is this is a regulator, a repressor of the matrix production. So in this mutant, you have a stiffer matrix. Okay, you have it here. The stiffness is larger. So what we see here, in fact, when we look at the wrinkles, the prediction is that the stiffness is going to determine the wavelength. of the pattern. Okay, so there are, we didn't do it, but there are already mechanical models of wrinkling, people doing material science, that they know that the wavelength of the wrinkles is going to depend on how stiff the medium is. And indeed, that's the case. The stiffer one, we see that the wavelength is longer. And the softer one, the wrinkle is lower. So this means that really the matrix, the cellular matrix and the depth is going to impact the three-dimensional structure by allowing all these compressive stresses to build up and by tailoring how the pattern is going to look like. And now I'm going to finish. The last one slide, I'm sorry. The last slide is, okay, if cell death is producing wrinkles, maybe we can engineer this. Maybe we can kill cells locally. The idea is, let's try to kill cells locally and see whether the wrinkles develop where we have killed the cells. Can you draw the wrinkles? And this is Munehiro did. And this is what he got. Okay, he took the biofilm, he just touched with a little tip, and this produces already death. So indeed, he got death here and in the little mouth. And after a while, okay, this is 16 hours. He induces the death in 16 hours. In 40 hours, he got, of course, the ring. This we cannot get rid of, and then the little thing here, which is a wrinkle, which is produced by the death. Okay. So anyway, that's it, and now I'm done. So I have tried to talk to you a little bit about how cellular decisions in bacillus are organized in time, how signals are integrated, and how is the population level organization determined by cellular behaviors. So I'm going to thank again all the people who really did all the work, and thank you for your attention. I apologize for going over time, arriving late. Very sorry. Thank you again.